Hi everyone. This is a recording of the talk I gave to the SMELT meeting on April the 30th. It's a re-recording due to poor audio. So by request, I've re-recorded the presentation and hopefully with better audio this time around. So I'll be covering how I've went about designing Nanodisc polymers from SMA to AASTY and the entire journey that has been. So the papers, let me see, there we go. So the papers I'll be covering is my first foray into styrene maleic acid copolymers, which was published in Biomacromolecules in 2017. And I'll follow up with a tool that was developed in part of that publication and how it took its own life and how I hope it will be useful to others. And finally, I'll wrap up with the story of how I use that tool to design better nanodisc forming copolymers based on acrylic acid and styrene, which was published in 2020. So the styrene maleic anhydride copolymer, which is the precursor copolymer for the styrene maleic acid, it's an interesting copolymerization. It's a radical polymerization with the two monomers that'll make a copolymer, which after hydrolysis will form styrene maleic acid by opening of the anhydride. And the ratio between the styrene and the maleic acid has a huge impact on the function of these polymers and their capability in forming nanodiscs. So what's special about this copolymerization is that the styrene has a much higher reactivity towards the maleic anhydride than it does to styrene itself. Approximately 20 to 30 times, this depends on specific conditions. And once maleic anhydride is the monomer that carries the radical, it can never homopolymerize. It'll only propagate by the addition of styrene. And so it continues. That means that depending on your initial monomer composition in your copolymerization, you'll likely end up with a very highly alternating structure. And if you have just a bit of maleic anhydride in excess, it will be pretty much fully alternating. This is a problem in terms of making nanodiscs with these copolymers in that you need a ratio of styrene to maleic acid of around 2:1. So that means that simply doing a batch copolymerization with these in a controlled radical polymerization will likely end up with some funky results, and I'll return to that later. But to get a styrene maleic acid copolymer at a 2:1 ratio, you have to do something special, and that is a continually stirring tank reactor. Such a reactor allows you to keep an approximately 30 times excess of styrene in the copolymerization at any given point. And when this kind of reactor is in equilibrium, it will quickly chug out the 2:1 styrene maleic anhydride copolymer in fairly huge volumes. So this is not something a standard lab can do. There are just a handful of these reactors in use in the world making styrene maleic anhydride copolymers. Such a reactor uses free radical polymerizations. And what this means is that you have a very poor control of the length of these polymers. Additionally, it can be very difficult to discern just exactly how tight your equilibrium was in your copolymer, in the resulting copolymer. So if you have a dispersity in your composition, that is, it is not a statistical distribution of 2:1 throughout the copolymer. That is very hard to discern if that is actually the case or not. So this has given rise to fear in batch-to-batch variations, in that some batches behave differently than others. So these are two important measures of which there is a poor control, or at least a poor measure of control in the second case in styrene maleic anhydride copolymers. So to put out a word on dispersity, so dispersity is the measure of homogeneity in length of the copolymer. And the SMA 2000 is listed as having a dispersity index of 1.7. What that means is that if you follow the x-axis on this particular plot, this will be the distribution of monomer lengths, and this goes from 1 to 600, assuming a average degree of polymerization of around 100. But the point of the whole thing is this is just not a single sample. This is a wide array of individual polymers with individual lengths. So for a polymer with a degree of polymerization of 100, the best possible dispersion you can have, and this is on the same scale, looks about this tight. So it's quite different. And this is not all, because this is not a weighted distribution. This is just, on this histogram, each polymer will appear with the same weight, regardless of how heavy it is. That is, if you have a polymer that is 5 monomers long, it will have the same weight in this diagram as one that is 1,000, but of course the one that is 1,000 will be much, much, much larger in terms of mass. So if you weight the distribution, this is what it looks like. So yeah, it is quite poor for a copolymer of 1.7. You have masses all over the place, and if you aim for 100, you'll see that a good chunk of your mass is well above 100 in terms of degree of polymerization. Whereas if you had a good control, well, not much has changed because, well, it's just a tighter distribution of polymers. So early on, when I wanted to look into this, I had the assumption that the length of the polymer had a huge impact in terms of either its capability of forming it or the stability of the discs, or somewhere in that direction. I was surprised that no one had looked into to figure out which polymers in the mixture actually formed the discs. So my collaborator, Thomas Laursen, made a series of, he made a big batch of nanodiscs with SMA 2000, and I took them into my lab and crashed out. He purified them with size exclusion chromatography, so you only had the nanodiscs and not the polymers that did not form the nanodiscs. And I crashed out the polymer and got away the lipids, and to figure out what distribution of polymers actually did the trick. So this is a size exclusion chromatogram, and the green area shows the raw SMA 2000 that has not been making discs. And the blue area is the polymers, is the elution diagram of the polymers that are purified from the nanodiscs. So this really showed me that, wow, this the size of the polymers definitely has an influence, as you would expect. And then we started working on the idea, like, couldn't we control the length and thus also the dispersity of the polymers? If we could do that, we could figure out what polymers work and hopefully understand why. And this would in turn lead us with the design criteria of making better polymers. So controlling length in a controlled radical polymerization, there's a few ways of doing it, but the one I mainly use is called RAFT. It's an acronym for reversible addition fragmentation chain transfer. It's simply an additive into a free radical polymerization. We end up inserting your 2 monomers in between the R group and the RAFT agent terminal group. And in this polymerization, virtually all the chains start growing at the same time, and preferably they should also stop at the same time. So they have like an equal lifetime to grow, and thus you can control the length of these polymers. What makes this interesting in terms of monomers being consumed throughout the polymerization is that if you have a change in your composition, say you start out with the 2-to-1 styrene to maleic anhydride composition, that will change as the polymerization goes along, as it's highly alternating. Because the copolymers continue to grow throughout the polymer, this change in the composition in the monomers is also reflected in the polymers, and I'll return to that very, very soon. But it raises the question, how do you actually? So that brings me to the Mayer-Lewis terminal addition model. So this is the model explaining, in the simplest terms, how a monomer is being consumed in a copolymerization. So if you have a monomer A, that carries the growing radical as it propagates, it'll have a rate of addition for it to self-propagate or homopropagate as an add-on to monomer A and there's a rate for adding on to monomer B. Likewise, we have the opposite scenario, or the same scenario. If monomer B carries the radical, it will have a rate of addition towards monomer A, or a rate of addition towards monomer B. So I have these two ratios of rates, and these are called the reactivity ratios. And if both of these are very, very small, then you have a very low addition to itself, and then you'll end up with a cross-propagating polymer. That is, it has a high degree of alternation. If there's no difference between adding on to monomer A versus monomer B, then it will end up being random. Or if it has a higher addition to higher rate of adding on to itself, then you'll end up with a somewhat blocky copolymer. But this is fairly simple math, and it has been known since, I think, the 60s. And it is very simple to computationally implement. And I wanted to use this tool to pick my initial conditions for my copolymerizations to try to hit some specific designs and copolymers between styrene and maleic acid. I have to say that, like, in terms of radical reactions, this is definitely not the only reaction taking place in the reaction mixture, but is the most important one in this scenario, and especially when designing polymers. So the way that the program works is that we have a preset monomer pool where you put in a number of monomers, and then you, with the reactivity ratios known, use a random number generator to statistically choose a value. So it'll either end up homopropagating or cross-propagating. So in this case, the growing polymer B added another monomer B, and the monomer B is subtracted from the pool. And this, of course, changes the statistics and the probability of what will be the next monomer to be consumed. And the program continues, and now it's on a monomer A. And again, this changes the statistics and what monomer is going to be added. So to do this sort of simulation, the only thing you need is you need to know the reactivity ratios. You need to know your ratios between your reagents, and you need to put a desired conversion in there. As in, like, do you stop it when 50% of the monomer has been converted, or 80%, or do you just let it go to 100? So with this tool, I started playing around with the styrene maleic anhydride copolymerization. And since this had a very big drift, and as I said very early on, that is very alternating, the easiest way to play around with a composition drift was to have a huge batch in the beginning, split it up in 5 parallel reactions, and stop it at different times. And so what you see here to the right is the very first output of the program, in that, like, if you stop it after 4 hours, you'll end up with around 60% probability of the monomers in any given position being styrene, and 40% being, oh, I got that the other way around. Oh yeah, styrene, and 40% of it being maleic anhydride. But as the maleic anhydride is being consumed faster than the styrene because it's alternating, the probability of copolymerizing in a maleic anhydride goes down as it continues on. So you have this changing composition in your monomers as your polymerization proceeds. So the polymerization, we stopped at around 16 hours. There was no longer any maleic anhydride left, and as the chain grows, there's just a higher and higher statistical probability that that any given monomer at the end of the growing chain will be a styrene, until it's just a complete little styrene tail at the end of it. So that was the idea, and I just played around with a lot of conditions. So if you end up with if you look at the bottom one, you have a styrene to maleic anhydride ratio of only 5 times as much styrene. It is almost completely alternating. It's almost 50-50 until the maleic anhydride is consumed, before it kicks up and ends up growing a little styrene tail at the end of it. Whereas if you end up with the start out with the styrene to maleic anhydride ratio of 20 times as much styrene as maleic anhydride, you end up with closer to this 6-to-6, the 2-to-1 ratio I aim for. However, in terms of controlling the length of this polymerization with the conditions shown, is like really difficult. It didn't go as fast in conversion as I'd hoped. So the monomers that were the copolymerizations that were most reliable were the ones with the monomer to raft ratio of around 200 and the styrene to maleic anhydride ratio of around 10. And this produces a series of copolymers with a varying extent of a gradient. So this is that series, series B, where we did an NMRs of each of them. And what was kind of funny, we saw at the end of it, once we started growing the little styrene tail, we started to see this shoulder grow at the end of it. We could also tell by the color of the polymerization. It shifted from an orange to a kind of pinkish hue. And this is likely a terminal effect. Like once the raft agent is right next to a styrene, it'll have a slightly different color. But this gives us a lot of polymers to play around with in terms of making nanodiscs. And what we found was that each of these gradients and their lengths had a huge influence in how well they worked. So very early on, it worked nicely with the B1, the very shortest one, as you would expect. As you hit B2, B3, we just included B3 in this case. It still works, but the discs moves earlier on in the iliogram, so they definitely get larger. And the time where we hit B4, kind of a lot of it is into the void, and at B5, we no longer solubilize the membrane protein that we were targeting, and the polymer is pretty much no longer functional. I forgot to say that this is a protein that we tried to solubilize into nanodiscs, which is coupled with the green fluorescent protein. So our readout here is fluorescence of the GFP. So with that, I want to continue on to talking more specifically about the program, as it's a tool out there for anyone to use. At this point, it can use up to 10 unique monomers at the same time. There's a lot of integrated analysis that allow NMR verification of your simulation. What I mean to say with that is you can get a readout of what is your monomer composition at the end of it. And if that matches, if you have a drift and the composition matches what you see in NMR at the end of it, then you probably have the simulated drift on your polymers as well. So what you need to put in is the target degree of polymerization of your polymers, the percent conversion as said early on, the monomer ratios and the reactivity ratios. And so the next one I'm going to show is a copolymerization with two monomers with a molar ratio of 5:1. Reactivity ratios of 0.05 and 0.02, so a fairly alternating copolymerization, and a number average degree of polymerization of 100 taken to 100 conversion. And these are kind of good numbers for the program to work in. So this is how it generates the diagrams I showed earlier on. And you see at the end of like fairly early on, the one with the lowest stoichiometric amount is being consumed. This is the blue line we see here, pops down. At the end of the polymer, there's just the red polymer left. And it browses through the different integrated analyses, but you can read more about those in the corresponding paper. So this is just a quick showcase of them. If you have 10% monomer, it's consumed fairly early on. You take it up to 50-50. In this particular one, as you would expect, in alternating copolymerization with a 50-50, alternating pretty much all throughout. And these are the lines that was in the first diagram before, the second one shown, and we've built in a crude way of implementing the dispersity index in the copolymerization. So you can change around with the how effectively your raft agent works in it. And this will give you different dispersity indexes. So it can give you an idea of if you have a pretty polymer or not and what that actually means. And yeah, I'll just browse to a few of them. So to wrap up the program, the theory is well known. Implementation has been very inaccessible for experimentalists. And the program Compositional Drift is a tool to traverse this gap, and the publication is out, and so is the tool itself. It's written in Python, but we also do executable, so it's fairly easy to get in for anyone to use. By the end of the whole SMA debacle, I had to think about whether raft was the right thing to do in terms of actually making good polymers, because I was limited in only being able to make copolymers with a severe gradient. And so with a single batch 2-to-1, you can make short functional polymers that has zero gradient, for sure of that. But we can't really control the size outside of that range in a single batch copolymerization. Now, I've seen Bert Klumperman has been working on a serial batch copolymerization. That's a way to do it, though that is technically slightly a bit more difficult. And regardless, there's an inherent statistical distribution of monomers, which is unavoidable with styrene maleic acid. And this is simply due to the fact that you need a 2-to-1 distribution in there, but you can't actually have it perfectly be 2-to-1 in the distribution along the polymer chain. It'll always be a statistical distribution. So it got me thinking, is there a simpler way around this problem? And this is when I remember the copolymerization reactivity ratios of acrylic acid to styrene, and looking at the polymer, it is very, very similar. So my thought was that a 1-to-1 copolymer between acrylic acid and styrene could do as good a job, or even better, perhaps, than styrene maleic acid. But it's kind of tricky to visualize a statistical distribution. Like how do you see something is more random than something else? I played around with my program, did some weird parameter changes to actually have the maleic acid appear as 2 acids. So this is why there are always 2 blues in a row every single time. That could give me an idea of how regular this polymer was as a whole. And what you see is that, yes, of course, as the reactivity ratios prescribe, the maleic acid will never come 2 in a row, as in there'll never be 4 acid units in a row down here. But the styrene is pretty much all over the place. The average is around 2, so that's what you'd expect. But there's just a statistical distribution. Now, the acrylic acid styrene copolymer is not as alternating as the styrene maleic anhydride copolymer. However, here we are not aiming for a 2-to-1, we're aiming for a 1-to-1. And so what I hope to see was to skew the population of styrene rundings, as in the number of styrenes coming in sequence, to be skewed as heavily as much 1 as I possibly could, while having a close as possible as a 1-to-1 styrene to acid ratio, similar to the styrene to acid ratio as you see in SMA copolymers. So here you see them together. It's kind of difficult to see which of these 2 is more random, and unfortunately mathematically it's also quite difficult to say, because while you can say that the maleic acid is perfectly alternating and get that distribution out there, it doesn't give the entire system justice. So I won't compare the statistical distribution between styrene maleic acids and acrylic acid styrene copolymers for the rest of this presentation, and neither do we do so in the paper. The mathematical model simply is not really applicable for it. So we went to Australia and did, after some initial experiments where we figured out, yeah, styrene acrylic acid copolymers will definitely work. We narrowed in on a few compositions around 50-50. We chose molecular weight ranges. We had a good idea would work from literature from other nanodisc forming copolymers. And these 4 polymers you see here were made in parallel and been running. These copolymerizations went fairly fine conversion. And the only thing I really want to highlight in all of this, except for the shorter polymer of the polymer A, the first one, is that there's very little composition drift in all of the polymers, except for at the end of polymer D, which has 55% acrylic acid. At the end of this, all the styrene has been consumed, and the occurrence of acrylic acid at the end of the polymer chain really starts to shoot up. Other than that, this is a very gentle polymerization to kind of play around with a narrow gap in styrene to acid ratios without having too much composition drift. So if you want to try a 40 to maybe and push it 30 to 70% range of acrylic acid, well, you can stop it earlier
and not have that much of a composition drift, and have a fairly alternating copolymer, but with variation that is forced upon the system by simply not having equal ratios of monomers. To the right is the different run length distributions of all of the copolymers, and you can see that once you add in more acid, as in the blue one, the times that styrene appears in twos, as in the run length of the styrene, starts to decrease. However, the acrylic acid run length starts to increase. So the system in the simulation, it behaves exactly as you would expect. Okay. So we got a bit of a help to kind of gauge the how alternating this copolymerization was, and we used a chemical correlation function and applied it to the program, and that was thankfully, with it being written in Python, was fairly simple to implement. And we could see that with the conditions run, including the conversions, the most alternating copolymer we had was polymer C. So the way this chemical correlation function is to be read is that a value of -1 is perfectly alternating, a value of 0 is perfectly random, and a value of 1 is a perfect block copolymer with equal lengths of each of the blocks. So for this, in this particular system, I would assume that the better we can get, the closer we can get it to -1, it would be better. So I was really expecting that polymer C with a ratio between styrene and acrylic acid of 1:1 would be the best polymer, in a given system. What we found that this was not the case. We found that the polymer A and B with slightly less acrylic acid in the system actually works a lot better. And now this is the same protein as we did in the SMA paper. And once you start adding in more acrylic acid and C and D, the efficiency in extracting this protein from the membrane starts to decrease a lot. However, all of them beat SMA 2000 out of the water. Now, this is not to say that these copolymers will beat SMA 2000 out of the water every single time. We aimed this specifically for mammalian membrane proteins because they are finicky and difficult to work with. And I expect that SMA 2000 works particularly poorly in these cells. Though since we were aiming for this protein anyway, this is a good thing to see. Another thing that's very special about the acrylic acid styrene copolymerization, styrene acrylic acid copolymer in comparison to SMA, is that it only has a single pKa value. So this is a Bjerrum diagram I did of all the copolymers. And you see that the pKa of the acid is around 6.8-ish for all of the polymers in comparison to SMA 2000. It has two pKa's that are very poorly defined. And now this is because of the dependency of deprotonating the second acid that's right next to the first acid. So the SMA 2000, as I understand it, works best around a pH of 8.3. So here you have some of the second protons of the acids being deprotonated. And what I gather from this is if 8.3 works well, the acrylic acid styrene copolymers should be able to have the same degree of charge at a lower pH, closer to around 7. So this is not something we've looked into at that point yet, if it has a better extraction efficiency at lower pH, but it's something that we really expect. When we took these particles into the cryo-electron microscope, they unfortunately were not as pretty as we would hope. We had them in abundance, but the resolution was just not that great. And for a talk on this, I'd have to refer to a later talk by Henriette Augsten, who will be talking a lot more about these copolymers in terms of extraction efficiency. So to sum up the AASTY, the optimized regularity of the copolymer is effective, but it's definitely not the only thing that matters. The acrylic acid, the acid to styrene ratio matters a lot as well. Raft adds new opportunities. Now, this is not something I got into in the AASTY paper, but in the SMA paper, we removed the terminal group of it under some gentle reducing conditions as well to generate a thiol. And that means that if you do a raft copolymer, you can add pretty much whatever you want at the end of the polymer in a very well-defined way. Now, this is not something you can do with the styrene maleic anhydride copolymers. Sure, you can add things to it, but it will be randomly throughout the polymer by opening up the anhydride, and not at the terminus in a one-to-one fashion, which raft allows. So this is also something you can do with the acrylic acid styrene copolymers. And that's the main thing. Like, this polymer can be made by raft, so you have a narrow distribution of molecular weights, and we imagine that this will give you a better control of your disc. It's just a neater and tighter system, and it can be made in any organic chemistry lab if you have a polymer chemist at hand. Whereas the continuous drain tank reactor and any innovation made on the styrene maleic anhydride copolymers are limited to like either the done material of the reactor or the people who are at these reactors. And raft adds new conjugation opportunities, and this is something I'll look into a lot in the future. And lastly, it's fairly tolerant to divalent cations, but for that I'd have to refer to Henrietta Auton's talk. And yes, as I said before, wider functional pH range compared to SMA. And lastly, I intend to functionalize the acrylic acid styrene copolymer because it's the nicest structure of the polymer that I can imagine and make. So having this alternating structure along the chain of it, I think, is a good scaffold to attaching a functional group of it and getting away from the inherent problems that are with having an anionic copolymer. I talked enough about the program, but this is the link to get it. The features I showed now are a bit, it can do newer stuff than what I've shown here. But yeah, shoot me an email if you want to talk more about the program. Lastly, I've started my own lab at DTU. It's named the Nanosmithery. And what I want to do there is leverage these advances in nanodisc technologies into tools of immunology. So in addition to helping develop tools for cryo-EM, I'll see if I can get something translational out of this. Lastly, I want to thank everyone who's been involved with this. Eric, my supervisor at Stanford, Joseph, my right-hand man at Stanford, Andy Spakowicz, who helped us further develop the program while I was there. I'd like to thank Professor Ting Xu for my time at UC Berkeley, Vincent Wu, who helped develop the initial version of the program, as well as Aaron Hall, my right-hand man at UC Berkeley. Also like to thank Yifan Cheng and Henrietta Auton for all the cryo-EM aspects of all of this work. And lastly, I'd like to thank Simon Harrison, who's the guy who initially got me thinking about reactivity ratios, and it has been a huge influence on the way I think about polymer as a whole. And everyone should read his work if you're into polymers, because he is a very novel thinker in terms of how we're thinking about polymers. And lastly, thank you for listening today.